So to kick off the kind of technique presentations, I'll start with an introduction to CT imaging. My name is Tamas Varga. I'm a material scientist here in EMSL, and I actually do CT imaging for MONet. My outline is I'll tell you about the fundamentals of X-ray computed tomography or XCT, then a little bit about the sort of information it can provide for us, and then end with some examples of the applications of CT data, CT-based 3D information. And then I have a slide of references for your records. So what is X-ray CT? It is a non-destructive X-ray imaging technique that is used to examine or visualize the internal microstructure of objects. Its output is a series of radiographs that are reconstructed by a computer into a 3D volume, hence the term computed tomography. And in CT, the materials are differentiated in the image data by their densities, actually the electron densities and their X-ray absorption. And this difference between the components results in different grayscale colors or intensities in the images. That's how we can distinguish them. X-rays interact with matter in a variety of ways. Some of it gets, the X-ray photons get absorbed, part of it gets transmitted, and some of it that gets absorbed leads to electronic excitations that results in applications, different X-ray techniques that can probe those, such as X-ray absorption spectroscopy, X-ray fluorescence spectroscopy, X-ray photoelectron spectroscopy, and some of it gets scattered, diffracted. You can do inelastic X-ray scattering techniques. But for CT, what we are interested in is the transmitted part of the X-rays that is left over after some of them get absorbed, and we detect those transmitted X-rays by a detector behind the sample. And what determines the transmission is the X-ray attenuation of the materials. So basically in CT, we differentiate the components by the difference in their linear X-ray attenuation. So the physical process of CT is that an X-ray source emits an X-ray beam that transmits through the sample, and that happens at many different angles because we rotate the sample during the scan. And the part of the beam that goes through the sample is intercepted by the detector, and the detector converts the photon signal, the photons that hit the detector based on what component they went through. That signal gets converted into an electronic signal, and then the electronic signal of each detector element at each angular position is used to generate a so-called sinogram in the computer. And these look like this. And these sinograms are then converted into a CT image via reconstruction step, which is typically either filtered back projection, that's the one we use in EMSL, or iterative reconstruction. And you can see in this figure how the number of projections, the greater the number of projections, the better CT data you can gather. So what does CT actually measure? As I mentioned, it is absorption based. So what it measures is basically the linear attenuation coefficient called mu, which is the percentage of the beam that gets transmitted through the components. So this basically contains the initial photon or X-ray intensity divided by the intensity of the beam after passing through that object, and then it's inversely proportional to the thickness of the material or the material traversed. And at the energies or powers that we use in CT, kilovolts, this linear attenuation coefficient is dominated by the so-called Compton interaction. And since this Compton interaction is proportional to the number of electrons or the Z number of the atom, we can say that this linear attenuation is approximately proportional to the physical density. So that's why you often hear it's a density-based technique. And I want to mention house field units, which are you see in the literature used by radiologists, often referred to as CT numbers. These house field units is basically it's a dimensionless number, standardized. It basically takes the radio density of water as 0, and standard temperature and pressure, and the radio density of air as -1000, also at STP, and basically sets a scale. This is a kind of a medical field scale. You can see that the different the bones and the different organs have different CT numbers associated with them, and we also use them, for example, in our way to estimate wet bulk density from MONet cores. In our formula, we also use the house field units. I want to talk about a few key terms that you encounter in the literature about CT imaging. One is the voxels. In photography, we have 2D pixels. In 3D, we have cubic pixels called voxels. And basically, the voxel size tells you your spatial resolution in the images. And the CT image can be considered as basically a voxel size pieces. Each voxel has a XYZ coordinate in 3D space and an intensity on the grayscale. The 3D images can be sliced and diced in different ways. We often refer them as image stacks because of that. These images are voxel distance apart. And so you can view the images as a stack, like TIFFs or JPEGs. Segmentation is the feature extraction, is the process of extracting features, such as here you can see a soy core where the porosity was segmented and false colored blue. I mentioned grayscale because the CT images are grayscale. It's basically just 2D images with intensity, one layer with intensities at each voxel, versus the RGB, the color images are multiple layers, such as red, green, and blue. The mesh is basically a polygonal approximation of the 3D shape. In this case, you see triangles that approximate the body of a dolphin. And these meshes are often used in the 3D data is converted into a mesh that is then used in models. And then the radiograph is, you're familiar with this type of medical imaging. The raw X-ray shows the air, low density part as light, and the higher density components as dark. When we reconstruct the images, we see that the colors invert. So now the lowest density part is black, and the higher density part is white. Let's talk a little bit about our EMSL system. In EMSL, we have a Nikon CT scanner, which is a large cabinet system. It's capable of housing larger samples. It has a point source and a cone beam configuration. This has implications about the spacing of the sample and the spatial resolution, as I will mention. In the scan, the sample is slowly rotated on the platform, which is placed between the X-ray source and the detector. And X-ray images or radiographs are collected at many different angles and then get reconstructed. This is a picture of the EMSL system. And this is my colleague Anil proudly posing with a MONet core. The MONet cores are scanned as received. They are 3-inch diameter, 1 foot long or tall. And you can see that also in the scanner. I'm going to go through these parameters of our system because they will describe the different options and considerations that you have in imaging. The we typically use the 225 kilovolt fixed tube. That is a little bit of higher resolution than the larger one, the 320, which has a higher penetration. But the 225 is applicable to most samples. We use that for MONet. So I didn't talk about how we generate X-rays. We don't have time, but they're basically generated by in vacuum electron beam hitting a metal surface and ejecting photons. In our case, currently we work with a tungsten reflection target, but we have an option to rotate that target to different metal surfaces so we can get different wavelength X-rays. Our detector is a flat panel plate, 40 by 40 centimeters, 2000 by 2000 pixels. And it creates 32-bit digitized images of the reconstruction. And for reconstruction, we use this Feldkamp-Davis crest or FDK home beam reconstruction because of the geometry of the instrument. The magnification that determines the region of interest captured and the spatial resolution can go from 1 up to 40. And the data collection really depends on a number of parameters such as exposure time, just like in photography, number of projections. You can get away with fewer projections if you have a narrow, tall sample, but the default is 3142, and the number of frames per projection collected basically results in higher signal-to-noise ratio. Typically, the MONet scans are around the order of hours, and you also have an option of continuous rotation versus shuttling mode, which is a stop and shoot between each little step, and that's what we use for imaging because it eliminates ring artifacts in the images. A few words about the resolution. So we distinguish spatial resolution and phase contrast, or our ability to resolve phases. The spatial resolution, as I mentioned about the cone beam configuration, depends on if you want to capture a whole object. You can see that if it's smaller, you can go closer to the source and get a larger magnification. So the sample size, the captured volume determines your spatial resolution. By rule of thumb, it's typically 1/1000th of the largest diameter of the sample. So for a 10 millimeter sample, you would have a 10 micron resolution, roughly. And but the resolution also depends on the X-ray power. You can see here that the beam focal spot size changes with X-ray energy. So we really have the maximum resolution, such as 3 to 5 micron at very low powers. Typically, we have 30 watts for the MONet cores. So that means that our spatial resolution cannot be better than 10-20 microns at those settings. And our ability to see phases, distinguish phases, such as the different densities, depends on the X-ray energy settings, and of course the density differences of the different components in the sample. I made this plot on some real-life samples to show you how the spatial resolution depends on sample size. So, for example, a 1-inch mini column or core would give you about 30 micron resolution. The resolution is taken as double the voxel size, because you need more than one voxel to describe a feature typically. And for a phone handset, typical size about 150 micron resolution. This would be more like a MONet core. Let's say a 20 centimeter tall object would have about 200 micron resolution. And you want to capture the whole size. So let's talk a little bit about the type of information that CT can provide. As I mentioned, CT provides a computer model in 3D space of the internal structure of the samples. You can look at CT as basically a 3D map of linear attenuation coefficients, where every voxel is registered to that location of the sample, and the measured intensity is related to the X-ray absorption in that location of the component of the sample. And so From segmentation, we can use CT to reveal, for example, root structures. This is a scan of a bean plant in the pot, and segmented the root structure, and this kind of imaging can be used to follow plant growth, how the root reacts to environmental disturbances such as CO2 content, drought, temperature. And because we know how many voxels build up the root, we can accurately calculate the volume, surface area of the root, diameter, and so on. This example is more related to MONet. In soils, we often reveal the soil pore structure, and so in this study, we're looking at how the pore structure determines soil wetting patterns. You know that the connected pore network of the soil directs the flow and transport of water, microbes, and minerals in the soil. So it is very critical or very useful to know the connected pore network from this data. I wanted to show you an example of an application of segmentation. This is a segmented data of a 4-component system. And I want to bring your attention to this histogram, which is typically more like a bell-shaped curve going from low density to high density. If you have peaks in it, then you have a better contrast, separation of the components. In this segmentation, each density will be just represented by a line. But so the lowest density would be the pores in this case, and segmented the pores. And then in green is kind of I think it's a polymer that partially fills up the pores. You see it's much higher density than the air. And then we have the matrix, the surrounding matrix has higher density. And we segmented these mineral containing or metal containing inclusions as the highest density parts. And as you can see, each of these can be visualized separately or together, and false colored and visualized accordingly. So the CT data can be represented in many different ways. We can, image-wise, we can provide digital radiographs, which is the simplest. But from full CT, we can provide images such as the 2D slice shown here. After segmentation, we can create visualizations of the different components. On the right, we applied basically transparency to the soil components, the soil matrix, and we only see the porosity. So we can provide those images, 3D perspective views, introduce clipping planes or slicing to highlight and emphasize components. And so these images can be basically any format, TIFF, JPEG. The 3D data are typically .wol or .raw format that can be visualized in or opened in the 3D viewer programs. But the images can be any format, and the animations created can be also AVI, MPEG, and other formats. And this example shows the visualization of a segmented root system. And in this study of a lily flower bud, we false colored the different components of the bud to highlight what the user was interested in. So a few examples of using CT data or application of CT. One is MONet. Obviously, in this is an actual MONet core showing the image data and the numerical data we provide for the project. We can visualize with the full CT, the full height CT, the different soil horizons, and the kind of a soil texture. And then from higher resolution data, in addition to the 2D slices, we can provide images of the pore network, total porosity, connected pores only, and those are also modeled using a pore network model. And numerically, we can provide not just total porosity, but volume, surface area, and diameter of every pore, each and every pore. And one can do further analysis on those pore spreadsheets. And then using the pore network model, as we'll describe also in the data tutorial in detail, we can derive flow and transport quantities or parameters. Another example is using CT data in models. So the pore space data can be converted into a voxel mesh, which is then used in flow and transport simulations, such as computational fluid dynamics modeling. This example shows how this data can be converted into a mesh, which is then used in a CFD calculation to simulate multi-phase flow. In this case, flow through a soil core. These codes that are often used for CFD, TETIS is a PNL developed code by Bill Perkins. CGS, developed at Oregon State University. Brian, who is a PNL PI, is a co-developer. And then the OpenFOAM is an open source CFD simulation code. These are often used with this kind of data. My next example is pore network modeling, which takes the pore topology from CT and basically creates a kind of idealized geometry of pores represented by spheres and the channels between them, the pore throats as sticks or cylinders. And this kind of modeling can solve flow and transport equations for the pore network. Codes are, for example, Aviso that we use for MONet. My colleague Anil and myself use Aviso in EMSL, and OpenPNM is an open source code for also pore network simulations. Ganchu Zheng, PNNL, is one of the experts using OpenPNM. In OpenPNM, we take the CT data, and it's a Python-based script. It can read in the 3D data into OpenPNM and calculate probability distribution functions, such as the distribution of pore size and pore throat size. And these are very useful for modeling diffusion in soil cores or soil samples. I want to finish with a few references that you can look at. A good summary of CT basics information can be found at this University of Texas CT Lab website that I like. The second link is our EMSL link to our CT scanner, also contains some good basic information about resolution and scan considerations. The third paper is the theory paper of the FDK reconstruction I mentioned. If somebody wants to get into it. I listed a pretty good soil science, XCT soil science review. Recently, we published a MONet perspective in the frontiers of soil science, which I recommend you to read. 1 year earlier, we published a paper that contained Aviso-based soil pore network analysis in geoderma on permafrost soils that shows some of the analysis that we do for MONet and more, such as pore throat sizes and pore coordination numbers. And then this European paper, recent paper of a soil structure library, is also a good read, showing a wide range of soil parameters derived from XCT. And next up is Professor Sasha Kravchenko from MSU, who will talk about applications of XCT for soil function in more depth. And I'm happy to answer any questions. We have about 10 minutes. Yes.
On an XCT scan, could you distinguish between like an organic fraction and a mineral component in a soil core?
It can. Often the limit is the resolution. So in a soil core, the soil organics are typically small in size. So as they aggregate to the larger size, if you talk about, for example, a MONet core, then this would be a higher resolution imaging, which would be roughly a 75 micron spatial resolution. if you have a huge organic aggregate that you can discern that works, but typically those are on the aggregate scale, and you don't see them very well. But from micro CT scans, we are able to see the particulate organic matter.
Do we have any plans to improve the spatial resolution for XCT through partnerships?
Yes, that's right. So we are partnering with synchrotron light sources such as the advanced photon source, where they have great micro CT capabilities to do imaging at micro resolution. So we have plans to supplement our lab-based imaging with high resolution micro CT on both from the larger cores, but potentially subsampling the cores and doing micro CT. We don't capture in these data, we don't capture cores that are smaller than 50, 70 microns. So there's still a lot there to capture. Yes.
This is perhaps related to the geoderma paper, but do these analyses have to happen at room temperature, or were you able to do those soils while frozen?
You don't have to happen at room temperature. We image at room temperature just because we don't have a well-developed capability to keep the cores at constant temperature. So the geoderma paper was on aggregates which were exposed to freeze-thaw cycles, but they were left to melt, thaw at room temperature for the imaging. That was just that was available or possible for that study, but for imaging in a frozen state, you really need to make sure that the temperature is kept constant, because especially in micro CT, it's very sensitive to small changes in the sample, causing blur. So even let's say 10-20 degrees temperature fluctuation can result in large enough changes. In micro CT, just see a blurring of the features. But we are working on a way to scan cores with cooling. Yes.
That's interesting to hear about their application for roots. The MONet cores are intact cores, right? So are we also scanning the roots in them? Or, I mean, it seemed like no from your presentation. Are they removed?
We are scanning them.
Okay.
Haven't been extracting them. So we're focused on the porosity. As you will see in the data tutorial, for the MONet cores, we focus on the porosity, but we do have plans to create workflows if necessary, and if the project problems and questions warrant, we can try to segment the roots as well.
Thanks.
Again, there is a resolution issue there, so if you can't really capture at this resolution fine roots, but if they are well developed, you can definitely capture them and even maybe model them, because they are also kind of channels for transport, but with a different hydraulic property than the pores. Yes.
Tamas, I was wondering if you can talk a little bit what type of data from the XCT will be available to the MONet participants. Is it everything you showed there, or is it a portion of it?
Yeah. So our data tutorial this afternoon will get into that, but basically image data and numerical data for not all, but these 4 parameters are included. We don't calculate 4 throats and 4 coordination numbers, but they can be calculated from the data. So these are, and then in addition to the pore sizes, we perform the pore network modeling, which gives us these transport parameters in different directions, XYZ directions. And then we also generate pore size distribution.
There is a question in the Zoom from Rich Phillips. So he writes, for those of us interested in plant-soil interactions, Are there ways to combine sensors so that rhizosphere processes like dynamic carbon allocation can be connected to root morphology? For example, measuring carbon flows after pulse, labeling plants with radioisotopes like 14 CO2 or 11 CO2.
Yeah, it's there are ways. So in that case, we would need to make the right samples, right, and make CT part of a kind of a workflow where you would segment the pores, segment the roots, model or measure hydraulic properties, and then you would subsample that core, get the sensor data, but also potentially those aggregate scale or rhizosphere scale data for imaging using multi-MS, nanosims, other microscopy, of course, and other techniques. So it is currently not part of the soil function call, but we have the capability to do that. And